Welcome to Section 8 of Metabolism. In this section, we'll be discussing fructose metabolism. Let's get started. This is the metabolic map provided in Section 1 of Metabolism. In this video, we're focusing on fructose metabolism. The fructose metabolic pathway can be seen right here. Let's zoom up on the pathway. This is a detailed figure of fructose metabolism, which can be found in Section 8 of Metabolism. Notice from the figure that the left side of the pathway, right here, shows the first part of glycolysis, which we discussed in detail in a separate video. The right half of the pathway, right here, shows how fructose interacts with glycolysis. There really are only two disorders that you need to be familiar with regarding fructose metabolism. Both of these disorders are shown at the bottom of the figure, right here, and correspond to enzymes marked by the stars. Before we talk about these disorders, let's quickly review the pathway. Notice that fructose metabolism can begin with sucrose. This is because sucrose is a disaccharide consisting of glucose and fructose. Notice the glucose is leaving as this is degraded. Fructose is then converted into fructose 1-phosphate by the enzyme fructokinase. This step is responsible for trapping the fructose inside of cells. Fructose 1-phosphate can then be split into glyceraldehyde and dihydroxyacetone, or DHAP. Both DHAP and glyceraldehyde can then be converted into glyceraldehyde 3-phosphate, which can continue down the glycolytic pathway to form pyruvate. Notice that some fructose can also be directly converted to fructose 6-phosphate by the enzyme hexokinase. This is an important alternative metabolic pathway that allows patients with a fructokinase deficiency to be able to metabolize fructose. Okay, now let's discuss disorders of fructose metabolism. A deficiency of the enzyme fructokinase results in essential fructosuria. This is a benign condition because fructose is not trapped in cells, and patients maintain the ability to metabolize some fructose through hexokinase, like we just discussed. You should know that both disorders of fructose metabolism result in an accumulation of fructose in the blood and urine. The fructose can be detected in the urine with a copper reduction test. The copper reduction test is a very non-specific test used to detect the presence of any reducing sugar. All monosaccharides, as well as some other carbohydrate molecules, are reducing. So this test will detect glucose, fructose, and galactose, as well as some other disaccharides. Obviously, if you're trying to diagnose a metabolic defect in carbohydrate metabolism, this test will not be very useful. However, it can be somewhat useful when combined with other tests that specifically rule out the presence of other sugars. Okay, let's return to the metabolic pathway. A deficiency of the enzyme aldolase B results in hereditary fructose intolerance, so fructose intolerance. This is a much more serious condition because fructose is trapped in cells and cannot be metabolized. Notice that ATP is required to produce fructose 1-phosphate. However, the fructose 1-phosphate cannot be further metabolized, so energy is wasted in producing this metabolite. The depletion of ATP in hepatocytes is responsible for the inhibition of gluconeogenesis and glycogenolysis. As these pathways are disrupted, the patient can develop hypoglycemia. So, decreased ATP results in decreased gluconeogenesis and glycogenolysis, which causes hypoglycemia. The accumulation of fructose 1-phosphate in the liver results in hepatomegaly. The liver can become fibrotic and patients can develop symptoms of liver failure, such as jaundice. Just like essential fructosuria, a copper reduction test will be positive. Symptoms occur following the digestion of sucrose or other compounds containing fructose, such as fruit and honey. Obviously, if compounds containing fructose are avoided, the patient will not develop symptoms, which is the treatment for this disorder. A previously healthy 8-month-old girl presents to the emergency department with nausea and vomiting. Her mother states that her symptoms started approximately 3 days ago. She also states that her daughter has been excessively tired. Labs reveal hypoglycemia. Her mother mentions that they recently began feeding her bottled food when her symptoms began. The most likely deficient enzyme in this patient results in hypoglycemia through what mechanism? From the question stem, you should notice that this patient has nausea, vomiting, and hypoglycemia, which started after the introduction of bottled foods. This is classic for hereditary fructose intolerance. From our metabolic map, we can see that hereditary fructose intolerance is caused by a deficiency of the enzyme aldolase B. Most questions about fructose intolerance will mention something about the introduction of a new food, hinting at the fact that the patient was given fructose, in this case, bottled food. So we're asked how a deficiency of this enzyme will cause hypoglycemia. And as we just discussed, recall that ATP is required to produce fructose 1-phosphate. However, the fructose 1-phosphate cannot be further metabolized, so energy is wasted in producing this metabolite. The depletion of ATP in hepatocytes is responsible for the inhibition of gluconeogenesis and glycogenolysis. So to answer the question, the depletion of ATP is most likely responsible for this patient's hypoglycemia. Okay, let's continue discussing the pathway. This is a figure of the polyol pathway, which can also be found in section 8 of metabolism. Notice that this is kind of a continuation of fructose metabolism. Some cells metabolize glucose through an alternative pathway by converting it into sorbitol through the enzyme aldose reductase. Sorbitol can then be further metabolized into fructose through the enzyme sorbitol dehydrogenase. This pathway is most clinically applicable to the pathophysiology of cataracts. The lens is particularly susceptible to damage due to the accumulation of sorbitol, especially when there is too much glucose in the blood. Under the conditions of hyperglycemia, glucose is quickly metabolized into sorbitol, and because the lens can form sorbitol faster than it can convert it into fructose, the concentration of sorbitol within the lens rises. So if we look at the pathway, we can see that if glucose is really high, it gets converted into sorbitol, but this cannot be converted as quickly into fructose. Therefore, sorbitol increases, resulting in the formation of cataracts. Because sorbitol is an osmotic agent, it increases the influx of water into cells. This results in damage to the lens and can be responsible for the formation of cataracts. So again, hyperglycemia results in increased sorbitol, which causes osmotic damage, which results in the formation of cataracts. Okay, let's do another question. An experiment is performed on two groups of mice. Mice from group A have a fructokinase deficiency, and mice from group B are healthy. The mice are fed fructose, and the rates of fructose metabolism are analyzed. How will the rate of fructose metabolism in mice from group A likely compare to that of mice from group B? Let's pull up the metabolic pathway of fructose to answer this question. So, mice from group A have a fructokinase deficiency. Mice from group B are healthy. We're asked how the rate of fructose metabolism in mice from group A would compare to that of mice from group B. As we discussed earlier, fructose can still be metabolized despite a fructokinase deficiency because of hexokinase. This allows the fructose to enter the glycolytic pathway as fructose 6-phosphate. Notice that fructose 6-phosphate is above the level of phosphofructokinase 1, or PFK1, which is shown right here. And this is the rate-limiting step of glycolysis. Because mice from group A have a fructokinase deficiency, all of the fructose must be metabolized through the rate-limiting step of glycolysis, which means these mice would likely have a slower rate of fructose metabolism compared to healthy mice. Mice from group B would metabolize a large portion of glucose through the more traditional pathway, which is below the level of PFK1. So notice that mice from group B are able to convert fructose to fructose 1-phosphate. This can then go to DHAP and glyceraldehyde, and ultimately to glyceraldehyde 3-phosphate, and then be converted into pyruvate. So this step essentially bypasses PFK1. This means the mice from group B would most likely have a faster rate of fructose metabolism.